Now we have our presenter from Seattle University here, who will give us a presentation about protein design, which I don't understand anything about. But it sounds super cool. And that's about Foo Café. We are always striving for something new, something which will be good for the future. And this is, if I understand it right, if the magazine Science and Nature is right, this is the fucking future. Sorry, the language. Thanks to Peter Neubauer. A big hand to Peter. So Peter pinged me and said, "This you need to listen to this guy. You have to meet him, and because he's doing some cool shit." And then Peter sent me some information. I said, "What? Biologic? No, I don't understand anything of this." But I took a meeting with Amijai. I can't pronounce it correctly.
That's okay.
Anyway, and here we are. And keep your ears open, because I assume you also need it. A big hand to Amijai.
Right. Thank you, Mikael. It's a pleasure to be here, and it's the first time of giving a talk in front of a dog as well, so I'm pretty excited about that. It's my first. So the title of my talk is How Protein Design is Going to Disrupt Your Industry. And my kind of lower objective is just to explain you what are proteins. If I made this, I think I made a kind of an achievement. So let's start from the beginning. What are proteins? Perhaps I will say something about myself first. I'm a postdoctoral fellow in the Baker Lab, University of Washington, where I mostly not with a robe or pipette. Perhaps this is a way biologists are conceived. I'm mostly sitting on a computer and doing code. Right, so let's start from the beginning. What are proteins? So, yeah, they are food, but much more than that. So I think the common kind of understanding of protein is something, an ingredient in your food. They are ingredients of your food because every living thing needs them to power and basically do the work in the cell. So they power and structure biology. So you can see here lots of proteins that were evolved by Mother Nature, and you can see they're super accurate and structured. And the reason for this is because each one of them is made to do a different function. And if you want to think about it, proteins are really nanomachines, right? So in the left, we see a machine as conceived by, I think it's Michelangelo, and on the right you have Leonardo da Vinci. Yes, sorry. And on the right you have microbial kind of propeller that is able to kind of push the bacteria in solution, and it's made of a lot of proteins that assemble to a larger structure, which provides bacteria with an ability to move. And if you notice the size, it's pretty small. So a nanometer is 1/1000th of a meter. It's very, very small. I think the tiniest thing we can see, like a hair, will be 100 micron, if I'm not mistaken, and this is much, much smaller. So we have a lot of those machines in a cell, and this is what allow us to live. Right, but this is a very complicated view of a protein, because you see this complicated structure, and it's doing this miracle of giving life and so on. But proteins are really simple. In fact, they're just a chain of blocks of like Legos. They're made of blocks. There are 20 blocks. So You see alanine, for example, and leucine and threonine. So there are different blocks, and there are 20 of them, and they create a chain, and the chain could be made of 200 or 300 block, and each block has a different chemical and physical properties, and because of this physical and chemical properties, they will fold into a structure. Right, and you can see here kind of a short animation that shows how kind of this chain is folding into, is looking for basically the place with the lowest energy, but it's slowly folding into a structure which is based on the sequence of the protein. In the nano world, the function of these proteins is derived from the structure. So I made 3 samples of what I call native proteins, which are natural proteins, and how their structure is deriving function. So on the left, we see an antifreeze protein. It's coming from a beetle, and it's protecting it in cold days from freezing. It could probably also protect your beer if you forget it in the freezer from freezing. But the way it does it, it has a very unique structure that organize the water molecules, and then it has a very similar structure to ice, and then it's able to bind the ice and limit its growth. And this, in this way, it's kind of, the structure is related to the function. You see an antibody, you probably heard about antibodies because of COVID and so on. So this is an antibody that evolved to bind basically a chicken egg protein. Not very exciting, but anyway, this is an example of this. And then on the right, we have an example of a machine that is generating most of the energy in our body. So without these machines, we won't be who we are. So it's extremely efficient machines that converts electron flow to kind of like cellular batteries. And it's really rotating, and in this way, it's able to kind of generate energy. Right, so I hope by now you kind of have an understanding of what are proteins, except being food, and you understand that they could be really useful for nature, for making life happen. But what is protein design? Right, so if you remember before, I showed kind of a sequence of a protein that folds into a structure. But up until very recently, if I gave you a sequence of a protein, very few people, if at all, would be able to predict in an accurate way the structure. So you get this sequence of 200 building blocks, and the sequence literally looks like this. And you wouldn't be able to say, okay, this folds into the structure here on the right. So this is named the folding problem, and scientists were trying to solve this problem for a very, very long time with some successes, but accuracy was always limited. So we couldn't go and take, let's say, a sequence of a bacteria in the deep sea and say, okay, this is a structure of the protein on these bacteria, because we didn't know to do this translation, right? There are good news about that, but I will get to it. So this is kind of the folding problem. So this is basic. If you want to do protein design, you need to know how a sequence will fold into a structure. Yeah, sure. Because a different sequence will. So maybe I will repeat the question so everyone here. So the question was, why does it matter to know how the protein folds? Okay, so let's say there are a number of reasons for this. First of all, let's say you're a clinician and you want to develop a binder for a protein on a virus. If you don't know the structure, you're unable to design something that will bind this structure. So that's first. And most of the time, most of the structures we have, if you go on databases, were experimentally proven. Now it's completely different. But up until a year ago, it was experimentally proven. So someone worked for 5 years in a lab, made a lot of the protein, crystallized it, got a paper in Nature, and published the structure of this exciting protein. So the work of multiple labs kind of aggregated into kind of a database where you hope that this mosquito or this bacteria had these proteins. So this is one thing, one reason why it's important. The other thing that if you want to design a protein, then you make a sequence, and then you want to know that it folds to the thing you want it to fold into, right? And like I said, structure dictate function. The more complicated question is, given that you have a structure, so let's say you decided that this is amazing structure and this is going to save the world from heavy metals in water. It's not, but let's say for the sake of it, this is a really important structure that you want to make. How do you predict the sequence that will fold into the structure? So this is kind of an opposite. Yeah, yeah.
Just a question regarding sequence.
Yeah.
That's not the DNA sequence. So what type of sequence?
Okay, so DNA, that's a good question. So DNA, I try not to dive into this, but if the question came, so DNA derives RNA. So there is transcription, and you get an RNA sequence, and then the coding sequence will translate into this code, which is basically just a long chain made of blocks, and each block have different properties. So you see each character is just an abbreviation for a chemical compound. So A will be alanine, I don't know, C will be cysteine, and so on.
So behind it is like the DNA sequence, which then gets translated.
Yes, so when you hear a gene, someone say, mostly, when you hear a gene, because there are exceptions to everything, but when someone talk about a gene, they talk about basically a sequence of DNA that eventually translate to RNA and then translate into protein. There are other things that DNA translates into, but mostly it's about proteins. Right, so again, going back to the design problem, so a structure can give us, we need to be able to say, we want this, okay, what sequence do I need to make this? And then obviously I can order the DNA and make it in bacteria or in yeast and so on. This is a more complicated structure because if you think about it, first of all, there could be multiple sequences that create one protein, and some of them can be good, some of them can be much better, so another level of complexity. And another thing is that you need to really understand that this is a structure you want, and this is a very, very complicated problem because we don't have intuition to this very, very, at least most of us, don't have intuition for the very, very small world, and things behave there really weirdly. So for example, I think if you would have asked people to think how a protein that inhibit ice growth looks like, I think very few people will guess that this is like the structure I showed you before. Okay, and like I said, things really changed recently, and this is probably the reason why I'm here. Because up until now, it was a very slow process. There was computational protein design. The lab I'm spending most of my day when I'm not in here in was working and making proteins before these breakthroughs, but the process was slow, and it was really hard, and it was tailored on specific scripts that were designed to make one protein. So really slow process that required expertise. But in 2021, there were 2 breakthroughs. It came in parallel. The left is Google. The right is Minkyang from my lab that now left to open her lab in South Korea. And basically, they use machine learning models to predict, to train them and predict from a given sequence, predict a structure. And this is a big deal because the accuracy of these models, definitely when you provide them slightly kind of a family of proteins, they know to predict protein in extremely good accuracy that sometimes outperforms even the experimental results. So this was crazy. And the speed of this is also quite amazing. Right, but why do we needed. So maybe I go back to your question. So first of all, we have very few experimentally proved structures. So let's say you want to save people in a developing country from mosquitoes that transfer a certain parasite. And you want to develop a protein that binds this parasite, but you're unlikely to find the smell receptor of these of this mosquito because no one was working on this mosquito. But now you can just go online and find the prediction, because we have sequences for a lot of proteins. 200 millions were folded by Google recently. So you can just go to your protein, see the structure, design a binder, and find a solution for this mosquito. And what you see here is a model of a protein that nucleate ice. Bacteria use it to kind of break leaves and plants material. So they kind of elevate the temperature needed to make ice. And this way, they kind of damage the plants and able to eat them. So this is kind of a very sophisticated. It's also used in ski resorts in Dubai to make snow. Just saying. Yes.
I don't know if everybody remembers, but the PlayStation 3 era was a program on PlayStation down folding at home. Folding at home. Yeah, folding at home. Do you guys remember that? Guys, is that like one of the features?
Right. I don't know what you refer to. There is a game called Fold It, which is run in our lab, and you can go to. Oh, yes. Sorry. Maybe I will repeat the question.
Hello, everybody. So my question was, if you guys remember the PlayStation 3 era, there was folded at home, if I remember correctly, that you can, if you were not using your PlayStation, you can just set it running and folding proteins, different proteins, and sending the data. I don't know to whom we were sending the data, but we did something at least. We helped.
You sent it to us.
Yeah,
in
hopefully for
Seattle.
good reasons. So that was my question, if it was one of the foundations, because
Yes.
there's multiple projects.
So
about
that's
that.
a good point. There is a game online you can go into. It's not. I mean, it's quite nice. It's cute. You can fold. You get a sequence, and you start by you see the sequence, and you try to use your own intuition to kind of fold the protein. And the idea was, we have a very good machine learning model right here. So the idea was like, okay, we have parametric models. Let's use human beings to learn how they get intuition and about how to fold it. And there was a nurse in Britain that used to work in the middle of the night and she had amazing intuition on how to fold it. But this was before the age of machine learning. So unfortunately, computers are slightly faster than us. And yes. Yes.
Sorry, folding at home was available on many platforms. It was crowdsourcing basically for doing computations. So you could have it as a screensaver on your computer and
Yes.
a lot of stuff. So yeah, but that was for trying to brute force with a lot of not so powerful pieces.
I think I will make someone in my lab very happy after I tell him that so many people know Fold It. Yeah, but there were attempts to use our machine learning model to try to improve parametric model, but this is another level of speed and ability. And I'm going to show you now, and I have here the links if you want to go and look at structures or use the notebooks to fold your own sequences. Right, so I'm going to show you one application of the machine learning simply because it's short of time and I still have a lot of things to show. But if you're interested in more, there are going to be opportunities to learn more about it. So you know how you go into Google or another search engine and you kind of write a sentence, and Google knows to complete your sentence before you even started. Well, you did started writing it, but you wrote a word or half a word and you already get your sentence. So this idea is that, like, given certain amount of text, can we complete the rest of the sentence? Could also be maybe implemented into a protein. So let's say we have a protein and there is a part we don't like. The part we don't like is the part that we masked here with XXX. And we have on the left the sequence and the coordinates of the atoms of these amino acids, the building block of the protein. And the right, we have another part of it. In the middle, we have a question mark. So they're not connected. Okay, so can we give it to a model and get
What about frog DNA?
frog DNA? From Jurassic Park, yes. So, okay, here I played around before I came, it's literally before I jumped on my flight. I was thinking about all the crisis in, sorry to mention it, but you know, east of Europe, and I was looking, well, do we have a protein that associates itself with uranium? So I found this a native one that you can see here in number 1, and then I said, well, can I take just the motif that binds a uranium atom here in 2? And then I said, well, maybe I wanted more than 1, so maybe I will take 3. And then I just gave it to the model and asked it to autocomplete and got 4, which is a complete protein. I'm not sure this will fold correctly, but given that I got 100 answers and I can make 100 proteins, then in about 20 minutes, I got from 1, which is a experimentally proven uranium binder, to 4, which is maybe a protein, but I have 100 samples of it, and one of them, based on our experience, is likely to have some affinity to uranium. So this was, I think, overall half an hour work. This used to be a whole PhD of someone. That's how fast. Now, another example to kind of illustrate how things are fast. For doing this mission, this kind of experiment or whatever, toying around with a script, I used generation 4 of this script. Generation 1, which is completely obsolete because we're in generation 4, was published in Science in July 2022. So we're now in August, and this script that was, or model that was published a month ago is 3 generations behind. This is how fast things are now moving. Right, so what can we do with that? So I showed you one example, but here are some examples from the lab. So this is an oligomer, kind of a ring that was designed by Natasha Adelman. She's an MD PhD, so just finished her PhD part, and she uses it to bind to the end of each part of this ring a medical or ligand or effector molecule to do stuff for therapeutics, right? So in the right you can see, zoom in, that you can see like the small rings forming in a very kind of nanometric view of this ring. Right, and you can also do fibers, and this could be useful for a number of things. You can also use them as detectors for stuff because they grow to a pretty big kind of structure, so you can see them in a normal light microscope, and you can, there are efforts now to do them like as detectors for stuff and to grab different peptides and so on for clinical purposes, but you can also use them for maybe fibers, maybe clothing, improve textiles and make them more sustainable. You can also do these kind of virus-like nano cages that are very useful for putting different combinations of molecules on them. We have an approved COVID vaccine based on this one, not this one, but something similar. Maybe the next one, not the next one. That was just approved in South Korea for injection, and it's presenting COVID epitopes. You can do a 2D array that forms these very nice honeycombs That's done by Ariel. Could do nanotubes that are huge and made of this. Imagine this. The software crashed if I made it into a nanotube. It was too many proteins. But you can imagine this ring extending into a tube, and this is massive. And you can literally run things inside this tube and use it for other stuff. So, and you have this another ring that was done by Basel. So, yeah, the most exciting one, you can do machines. So here you see an example that you have a kind of a rotor that is moving, and you can connect energy source for this, and then it's literally moving, and the measurements of the structure moving. So a small nano machine designed completely from scratch. On top right hand side, you can see the pictures of the real thing as constructed. So this was done by Alexei. Okay. So how is this going to affect you? So it's all nice that we play with proteins in the lab, but how is this going to affect protein? And I will try not to bore you only with medicine, right? Because normally when we talk about biology, we talk about medicine. But I think the main application is actually in other things, not medicine. So in medicine, this technology is already very disruptive. So here I gave 3 examples of what you can do with this. So on the left, you see a detector. Basically see a switch. The protein was designed to open only when it's meeting a certain signal or a peptide or recognizing something. So you can think of a death signal that is, yes?
Hi, super curious. I understand it's a very complicated thing, but how do we go from a picture on the screen to something that you can like inject in my shoulder?
Oh, so once you have a sequence of a protein, then all you need to do is to translate it to DNA, which takes, I don't know, 5 seconds. And then you have now DNA printers, so you just order 1 or 100,000 sequences, and then you put them into bacteria or yeast, and you make grams of it. It's still quite expensive, especially for clinical applications, but if you display it on yeast, for example, if you don't need it for clinical applications, then it's much cheaper. But for clinical purposes, you need to clean it from toxins, and you have to go through lots of assurance. But actually, to get the protein, it's quite straightforward. You go online, order your DNA, you get a small piece of paper with your DNA, you throw it on bacteria, you get your protein. It's that fast.
I'll try later on.
Yes.
I'm curious about how fragile this structure and in which environment it works. So is it easy to break these proteins and these folds?
Okay, that's actually a good question. So proteins could be, like I said, you can have several sequences for a structure. A sequence will fold into one structure. Sometimes you can have a sequence folding for 2, but this is an exception. But mostly a sequence fold into one structure, but one structure could have a number of sequences. And you can tune the proteins to be very stable. So we have some proteins that you can boil and they stay stable, or you can leave on your table for a year and they will still stay stable. But some proteins will fall apart very quickly if you elevate the temperature. The main problem with physiological environment is other proteins that want to either attack or kill or destroy proteins, because we were evolved to attack anything that is slightly different from us. And yeah, there is this is the most evolved system in our body. However, you can even design the sequence to be very similar to human proteins. So you can take motifs only from human proteins when you are asking the model to kind of, when you design what called the loss function, you can ask the protein, like the model to generate the same structure, but only with human sequences. And then you reduce kind of immune problems. In terms of stability, you just you need to mostly design proteins that are super stable. Okay, so I will go quickly through those. So we have on the left a detector. So something that happens, you think about logic. So you can have an AND detector, an OR detector, a NOR detector. So you have an action happening only upon a certain signal. You can have this very efficient cage that you take an antibody, any antibody, let's say against COVID, you throw the cage component in pink, and they form like this monster structure with antibodies pointing from all direction, which in nature means that it will bind tighter to the target. Or you can design mini binder to almost any target and neutralize it. So you remember I mentioned that now you can find the smell proteins, smell kind of receptor of a mosquito in Africa. So kind of very quickly now you can design proteins that will bind it and neutralize it. And you can think about application of this for medicine. And here I said like the COVID mini binder here, I think it's an example. Right, but let's talk about other things because I guess most of you are not in biotechnology or medicine. So let's talk about something that is going to affect most of us, which is taste, right? I think most of us drink things, soft drinks, and not that healthy. So the way we make soft drink now is that we take water, we add sugar, and we add lots of other molecules that mostly our body is not familiar with, and it has different complication. Some blame it for a lot of weight problem around the world. And in nature, we have these native proteins that are sweet. They are giving exactly the same taste as sugar, but they're proteins. They're just faking sugar. They can even feel sweeter than sugar. We also have taste modifiers that will make you think you're drinking sweet when you're drinking sour, or drinking sour when you're drinking sweet. And this is done by nature. So now that we're able to create libraries of 100,000 proteins that bind in different kind of strengths to our taste receptor, or you can imagine smell receptor, although this is kind of a more complicated task, then you can combine these, do combinations to do different tastes. Now, I think to hit exactly the taste of an orange juice will be difficult, but why not just generating new exciting tastes with waters that are completely healthy? I mean, all you drink is water and a little bit of protein. So obviously there are other ideas in the food industry, right? I talked about the mosquitoes. So, you know, nowadays, kind of, if you grow vegetables in your garden, or you have flowers, or you're a farmer and you have your kind of field, you have a kind of 5-kilo hammer. You don't want insects, so you put this thing. At best, it will be organic, and it will kill everything, right? From bees to insects that we don't want them there. What if we make, instead of doing this, we just make proteins that kill or even deter specific insects that we don't want in the field? So instead of destroying lots of poisons somewhere, you design proteins that will scare away exactly, for me it's mosquitoes, sorry, and like, that's my my thing. But what if we use proteins for this? Right. So what is the next step? How much can we take it further? And this is my favorite part, because this is kind of the thing I'm working on, so I'm a little bit biased here. So basically, proteins are very good in organizing other material. When you look at a plant outside, or a fly, or a crab, it's mostly made not from proteins. There are other molecules like cellulose and chitin, and also in organics, if you look at the shells. And our bones have some proteins, but they have calcium carbonate as well. Protein knows to organize other material. What if we can make proteins organize other materials for us? So here I gave a few examples. You can ask a protein to nucleate another material for you, so grow another material in a specific shape, you can force a naturally growing shape to grow into another shape by blocking one side of it. Or you can take things like diamonds, for example, and put them in a structure together. So this is kind of what I'm working on, and this is not my work. This is 4 billion years of evolution. So on the left, you can see mirrors that are designed to reflect light underneath the water and create an eye in the water. You see glass, silica, organized by, I think these are diatoms, so kind of bacteria, that is coated instead of like the normal coating in glass. And on the right, you see magnetic bacteria. So bacteria that generate a line of nanomagnets. And there are several explanations for this. I don't know if I buy them, but yeah, there are a number of explanations. Part of it is to find the north. I don't think that this is the case, but anyway, they do make these nanomagnets that are extremely small and organized. So what can we do with that if we do this? So how about, for example, doing computing with proteins? Can we organize atoms in accuracy that will allow us to move light in a very specific way? So moving the light atom by atom. So this is kind of a prototype of design proteins that organize a lanthanide. A lanthanide is an element that it's a group of elements that have very unique properties and very unique fluorescence and so on. I'm not going to bore you with this, but if you organize them into a very specific structure, you can make the light move between them, and you can get very interesting phenomena in terms of photonics that are thought to be useful in photonic computing, right? And can we really do this? So this is like a sneak peek into what I do. So on the left, you see a fiber protein I designed. So you see a fiber with a small twist. This is composed of thousands of proteins that are organized into this fiber, and these are actually two fibrils that are coming together. When we mix them with the ingredients to form a crystal on the right, in a kind of a very low concentration, you can see these structures of organized material forming specifically on these fibers. So maybe we're getting there. There are a few more examples, and maybe this is going to be a future, and just another example of what you can do. So we talked about textile, and if you put on a fiber specific mineral, you can get really interesting properties. So this is just a joke, but if you add a certain compound, you can change the kind of the wavelengths by which heat is being irradiated and send your heat directly to space, which will be nice. Right, so let me wrap up. So why Sweden, right? So I think you can say on a number of countries, or at least number of countries will claim that they are the leading European innovation hub, but Sweden is definitely out there, high out there. I think this region is very famous for being a biotech hub and also gaming hub, and has strong coding and machine learning. I think people here really care about transition toward a sustainable economy, which could be a very good reason to do things with proteins, because this is a way to do things that can break like in nature and be produced the same way nature is producing them. And finally, there is some very unique legal infrastructure in Sweden that means that if you're a small startup and you only have GPUs and a computer and you want to make a protein, then you can just talk to a professor over fika and start collaborating the next day. And this is very unique, because in other countries it will take you a year of negotiation with the university to do exactly the same thing. So if you're a group of developers, you can very quickly, and you have a great idea and you tested it, and online, like on your computer it's working great, you can move it to the lab very quickly. And this is unlike other places, which I think it's great. Right, and a plug. If you're interested, if you find this amazing. Then there are going to be a few workshops that are going to dive into these and really teach how to use this machine learning model to design your protein only with your laptop, and probably you need a GPU. The first one is on the 26th of September. You can already register on the Foo Café site if you're interested. There will be more, one in October and one in November, that even if you came to one, it's good to come to others, because each time there will be another model. You can also come to the second or the third one if you haven't been to the first. And then in January, there's going to be a hackathon, if anyone is interested. And thank you. Thank you to my mentor, David Baker, who practically invented the field of protein design, and all the teams that I showed some of the work. Peter and Pale Blue Dot that helped me to connect to people, and Foo Café, of course, and Michael. And that's it. And thank all of you.
Thank you, Amijai. You need to give me a hug. All right. No, a big one. So, no, a real one with both hands.
Both hands.
All right. Thank you. Thank you very much. And yeah, it twisted my head around, and I'm not really the brightest person here, so I assume there will be tons of people turning up at the workshop. And some other thing you told me, that the hackathon will probably be the world first hackathon on protein design at Foo Café. Yay! All right, now we are continuing to party, and there are more drinks and more people you haven't met that you could meet and just hang out here. And Aisha, she's opened the candy machine, so you could play candy, not Candy Crush, but but Candy Get, what? Yeah, well, Candy Crane, it's called. All right, thank you. I'm so happy to see you all here. I love you all. All right, let's go party.